This is a paper on mRNA vaccines titled The Dawn of mRNA Vaccines, the COVID-19 Case. This is a 2021 paper. So at the basic level, the COVID-19 mRNA vaccines consist of synthetic mRNA strands encoding the COVID spike glycoprotein packaged in lipid nanoparticles to deliver mRNA to cells. In other words, the vaccine causes your own cells to produce spike glycoproteins that are specifically coded through the mRNA, and these fragile messenger strands are packaged tightly inside of fat. Tiny little fat particles protect them. So for a little history, the older school basal vaccines trigger immunity by injecting weakened or wholly inactive pathogens. So essentially, they would either damage the pathogen or just give you a dead version of it, and then your body would generate an immune response to that. However, more recently, for things like human papillomavirus, HPV, and hepatitis B, or even the seasonal flu, they no longer contain intact virus particles, but rather purified or recombinantly produced viral proteins. So we moved away from having an inactive or weakened form of a disease to producing just the proteins from that pathogen so that your body can produce antibodies to those proteins, to now with mRNA vaccines, they don't even contain proteins or even the whole virus particle, but instead consist of messenger ribonucleic acid that can code for the protein itself, using your own cells to make it fresh. Now with mRNA, one of the earlier problems was that your immune system would recognize it as a foreign pathogen and target it and destroy it before your body could actually create antibodies. So scientists developed a technique called in vitro transcribed mRNA, which replaces certain nucleotides with those that are naturally occurring, which dramatically reduced the amount of innate immunity activation, so your immune system wasn't capturing and destroying the mRNA before it could be transcribed and produce the target that you wanted to have the antibody response to. With the benefits of this being that it enables a much higher protein expression and has a better safety profile. And both BioNTech or Pfizer and Moderna have COVID vaccines that are based off of this method. The next advancement is in regards to the delivery vehicle itself, which is these lipid nanoparticles. These work by protecting the mRNA upon administration of the vaccine, and during the mixing step, the mRNA that is negatively charged and the lipids that are positively charged naturally create this pocket, where a bunch of tiny fat particles surround the outside of the negatively charged mRNA, which improves both the stability of the delivery vessel for the mRNA itself into the cells, as well as promoting safe transport through the endosomal layer. And to underpin how fast this technology is and how fast it can be produced, 42 days after sequence selection from the genetic sequence of the new COVID variant in China, the first clinical grade batch of Moderna's mRNA vaccine was ready. The third challenge to overcome was that once you produce these spike glycoproteins from the mRNA, you don't want them to bind to anything and cause problems. So the genetic sequence can actually be altered slightly to stabilize the spike glycoprotein in a pre-fusion conformation, which means that it won't bind to anything, which is great. And the major advantage of the mRNA approach, where you're having the person's actual cells produce it, means that you're going to get proper protein folding every single time, and therefore generate a highly accurate target for the immune system to generate antibodies to. Now, one of the big things with the COVID vaccine that we mentioned earlier was changing it to match mRNA that's produced inside the body itself, which is slightly altering the chemical structure of uridine to an N1-methyl pseudouridine. And this is one of the major things that was awarded the Nobel Prize. Now, what is the goal of the mRNA vaccine? Well, the primary focus is to trigger B cells to promote the induction of neutralizing antibodies, which are those memory cells, and the goal is also to generate these CD8 T cells and these CD4 T cells. So in essence, you have the injection into your muscle, these get transported down into the endosome layer, which then degrades that outer lipid coat, allowing the mRNA to escape, head to the ribosomes where it will be produced into the spike proteins. And then these spike proteins, these spike antigens, will produce an antibody response of both those B cells and T cells. And the goal is for this to happen in the lymph nodes, the lymphatic system.